In this lab, we were exploring the enzyme catalase, which is found in most animal tissues. Here we have liver, and how it reacts with hydrogen peroxide. The enzyme helps speed the reaction of the breakdown of hydrogen peroxide into water and oxygen, and we can view the oxygen as it produces bubbles in the reaction. So what we're going to do is we're going to add 3 mL of hydrogen peroxide to each one of these liver samples. One is at room temperature, one is warm, and one is cold. And we can watch the bubbles to see how quickly the reaction is occurring. So starting with room temperature. The bubbles are oxygen. Now we're going to go to the warm liver. So you can see there's actually more bubbles being produced. And finally, we're going to go to the cold sample. And the cold produces some bubbles, but definitely not as much as the warm. So this enzyme has an optimal temperature of around 98 degrees Fahrenheit, which is about the temperature of the body. So it works best when it's warm. The other thing we learned in this was that as the reaction is taking place, it produces heat. Okay. This is known as an exothermic reaction. The other thing we looked at was whether the enzyme was reusable. So notice we have our room temperature. I'm going to take this out and we're going to place it kind of in a cleanish area. And then we're going to add hydrogen peroxide again to see if it was reusable. So here we have this, and it's still bubbling, indicating that the enzyme is not used up in the reaction and can be used over and over again. We could keep adding hydrogen peroxide and it would keep bubbling. Now you might wonder what's in these bubbles. The bubbles are mainly water and oxygen. Since the enzyme is over in the liver, if we added new liver to that stuff that came out of it, would we expect it to bubble? Well, let's find out. Now we put liver in there. It's not producing any more bubbles. Why? Because the hydrogen peroxide has already been converted into oxygen and water.